So maybe we should start then. So welcome everyone to today's Oh, is it working? And we're connected. All right then. So everyone welcome for today's seminar. And it's my great pleasure to introduce Elisa Dominguez-Hüttinger, who is our speaker today from the National Autonomous University of Mexico, but currently spending a few months at the Wissenschaftskolleg zu Berlin in Berlin. And by the way, I should take the opportunity to make a pitch for the Wissenschaftskolleg. I sent around call for applications a few weeks ago. Deadline is on Monday. Highly recommended for senior postdocs, young group leaders. So Elisa is a biologist by original training, but very early in her career, she already got interested in a mathematical modeling approach to things like onset of disease and various switches in biological systems. She did her PhD at the Imperial College in London, where she worked on mathematical modeling of atopic dermatitis and proposed mechanisms for the onset of disease, which was later, through a collaborative visit, verified experimentally in Japan. But she also applies the same approach, inspired by dynamical systems and control theory, to various problems such as how do we get pneumonia from a bacterium that's usually harmless, or even switches in ecological systems. So with that, I'll hand over to Elisa. Welcome.
Thank you. Hello. Sorry, there was a mistake on my phone. I pressed the wrong button. Sorry about that.
Please use this mic.
Yes, just now. We just got working now. Thanks a lot.
Online people, shut up.
So we have to be robust to perturbations, so it's okay. So when things go wrong, and they can go wrong in many different ways, many things can happen. So maybe the most obvious one is that we could get infection, right? So we don't have this regulation anymore, so we can get an overgrowth of pathogens in our system. But we can also have an overreaction of our immune response, leading, for example, to chronic inflammation or accumulated cytokines. And we can have even more severe phenotypic alterations, which are, for example, responsible for malignant transformations of the tissues, for example, in cancer. Something is going wrong. So why do we study this kind of tissues from a mathematical perspective? Today, I will argue that we can use mathematical models to address current and very urgent challenges in understanding, preventing, and reverting epithelial tissue diseases from a biomathematical perspective. And in my talk, I will go through 4 questions. So it's a 4 sections or a talk in 4 acts. First, I will talk about how we can use mathematical modeling to shine light on pathogenic mechanisms, which are often unclear for this type of diseases. Second, I will explain how we can use things like bifurcation analysis to clarify what is the effect of different possible combinations of risk factors, such as genetic and environmental alterations, on the maintenance and loss of homeostasis of these tissues. Then I will very briefly touch on a new field that we are working on in my group, new field for us, which is trying to address this problem that this kind of dynamical diseases show a sudden progression from a clinically subtle state to a severe disease stages, which are very hard to revert. And finally, I will touch on some work that we have done to address the fact that most of the times when things are already on this bad side of the spectrum, when we try to use treatments, such as, for example, antibiotics or corticosteroids or even chemotherapy, these treatments have very often negative side effects associated to them, and they might also be ineffective for some patients. So to address these 2 questions, we use mathematical modeling to optimize and personalize treatment strategies. So today I will focus on 2 specific types of epithelial tissues. First, I will talk about skin, more specifically about epidermis and its diseases. I will mainly focus on atopic dermatitis, but currently we are expanding our model to also encompass more severe diseases such as psoriasis and carcinomas. I will also talk about airway epithelium infection models, specifically about 3 of them. The first, Savolj also already mentioned, which is infection by Streptococcus pneumonia. And I will also very briefly talk about 2 new models that we have been developing in our group, trying to address COVID and tuberculosis infection. So part 1. We want to use mathematical models to unravel disease mechanisms. So we want to be able to go from a description of the data to a prediction of what is going on. For that, we start the very first and probably most time-consuming part of this type of research projects is to gather data. So we read, talk, read. It's almost like, I feel a bit like a psychoanalysis sessions, like really trying to extract even the unspoken parts of data. So we gather data, assemble it together, integrate it, and then formalize it into a system of equations. So in general, we use nonlinear dynamical systems that look like this. And more specifically, we use ordinary differential equations, but to which we very often add some discontinuities to mimic, for example, treatments, and also to allow us to account for timescale separation, which is typical of this kind of systems in which we have fast biochemical processes coupled dynamically to slower tissue level processes or dynamics. Sometimes also to address questions like what are the early warning signals for disease deterioration, and also how robust a system is, we can use a stochastic version of our models to explore, for example, how variance changes when we change given parameters. So when we start our work, so this puzzle making, so normally we start by sketching cartoons. Like this morning, I had a very great time with your beautiful blackboards. And this is the kind of things that we do. So we sit down and just draw the cartoon of what is going on. I will show you only very briefly what this represents. No need to go into details today, but if you have questions, I will be, of course, happy to answer afterwards. So this is a cartoon of an epidermis, the uppermost layer of the skin, where we basically have three layers: a basal layer, which is non-differentiated but proliferating, a granular layer. So I'm skipping all the other intermediate layers of keratinocyte differentiation. A granular layer, which is the uppermost viable layer with terminally differentiated cells. And then we have the skin barrier, which are cells that are already dead, but which still have an extremely important functional role in forming these lipid layers that hinder the excessive exchange with pathogens and water. So this system is tightly regulated. And when things go wrong, for example, when the skin barrier becomes leaky and pathogens come in, they can come in contact with the viable cells and mount immune responses that will have at least two effects. First, as you might expect from a maybe naive perspective, immune responses might counteract this pathogen load, for example, via secretion of antimicrobial peptides. But there is also, and I would claim that this is always the case in immune responses, a negative side effect on the tissue given by this inflammatory or immune responses. In this case, particularly, every time immune responses are activated, also the formation of the barrier is decreased, both by hindering the formation of the lipid layer and by overactivating proteases that will further chop down the barrier. So this is sort of a paradox. So when we study this kind of systems, there are other things I will skip now for the sake of time. So, but I want to briefly stop at this paradox. So when we look at these kind of systems and we see that when we have a perturbation from the environment, in this case, a pathogen, and two opposite things can happen. So on the one hand, we get this activation of immune responses that decrease pathogen load, but on the other hand, we will also have a burden on the tissue. So our claim is that in order to maintain homeostasis, we need to have a balance between these two feedback mechanisms. And to address this question, we then represent these cartoonish interactions into a regulatory network where this and the other variables I did not mention now are represented can then easily be represented as model variables. I want to point your attention to these switching gates. They will be important in my talk because they represent the abrupt on and off switching of phenotypic responses, for example, immune responses to challenges which are also history dependent. So what do I mean by that? When we have our pathogen here in contact with the skin cell, at the beginning, the skin cell will not respond. So no phenotypic response, no response, no response. And suddenly when we reach the threshold, suddenly we will have a very high response to this pathogen. And this response will persist even if we decrease the load below, not until we reach a threshold, which is strictly below the first one is that the response is now turned off. So this kind of hysteresis behavior, which also from my talks to you this morning, I know that most of you know very well, is very, very important in maintaining homeostasis in this kind of systems. So next, we construct also the control structure to really try to clearly pin down what are the feedback mechanisms that regulate, in this case, this external perturbation, which is the pathogen load. So once we have our regulatory network, then we can finally formalize it. And what we do is we write down a system of differential equations. In this case, we have 4 variables, 4 smooth dynamical variables representing pathogen load, barrier function, antimicrobial peptides, and adaptive immune responses. We also have these 3 switching functions, which represent this bifurcation diagrams here in a phenomenological manner to represent this abrupt on and off, turning on and off, or phenotypic decisions in response to changes in dynamically changing microenvironments. So those are the immune responses and also adaptive immune responses and the response to keratinocytes to changes in barrier function. So together, this gives rise to piecewise smooth differential equations, which represent a lot of opportunities, and some things are easier to analyze than in higher dimensional smooth dynamical systems, but they also represent some challenges, which I will talk about you today. And once we have written out a system like this, then we can, if we know beforehand, identify already some of the bifurcation parameters. If, for example, there have been genome-wide association studies pointing out specific genetic variants that might act as genetic risk factors. In this particular example, we have these 3 key risk factors. So one of them is clearly because I've been talking about this, the amount of external pathogen load, so how much you're exposed, our skin is exposed to pathogens such as Staphylococcus aureus, but we have also the innate or the permeability of the barrier, which can be modulated by mutations in a gene called filaggrin. And we also have the strength of the innate immune response, which can be genetically modulated by changes in expression of receptors such as TLRs. I just want to very briefly comment on, so that you don't feel that these switches are esoteric. So in fact, today I'm focusing on multiscale models where we already assume that we have these switching functions. However, a parallel or nested line of research in my group looks more carefully at what happens inside a cell by looking at the regulatory networks that give rise to such abrupt on and off phenotypic decisions. And what we do here, and this in particular is work from an undergrad student of mine, we assemble the gene regulatory networks that are known to be, in this case, responsible for keratinocyte differentiation in response to barrier damage. And then, long story short, I'm skipping many details here, but we construct a mathematical model here, a smooth nonlinear system of differential equations. And when we analyze through bifurcation analysis, we see that indeed it shows this abrupt on and off behavior, which mathematically corresponds to saddle node bifurcations or by stability. So once we have our mathematical model, the next step is to proceed with the calibration. And here, again, I want to just stop one moment to emphasize the fact that we put a lot of effort in integrating multi-level data sets. So I come from Mexico, where the experimental resources are very, very limited. So we really make our very best effort to squeeze out all the possible information from any type of available experimental data. And more specifically, something that we aim to do is to try to explain in vivo observations, which are the most valuable and the most expensive, both in terms of money, time invested, and also, of course, the lives of the animals, in respect to the regulatory mechanisms that very often can be described experimentally by in vitro experiments of subsystems of this whole in vivo system. So here I will use a second example, the example of the airway epithelia. It's a similar system to the skin. I will not go to details, but again, we have a bacteria, in this case, it's called Streptococcus pneumonia, which comes in contact both with barrier and with immune responses. In this case, it's macrophages and neutrophils. And again, we have this beautiful balance between positive and negative feedback loop, where we can explore if that is responsible for maintaining homeostasis. So for this particular model, we use data from dynamical data from animals, but we also use a whole set of other experimental data sets, including simply ranging from simple growth curves of bacterial cultures in different culture media, and also this so-called killing assays to assess how strong the which quantify the strength of immune responses against this given pathogen, and this called transmigration assays, where epithelial cells are co-cultured either with immune cells or with pathogens to see this interplay between bacteria or immune cells and barrier. So this gives us a lot of information, even quantitative information, that we can build into our model. I just want to say here, however, that to do this, we have to spend a lot of time in constructing a standardized database, which is not an easy task. And well, ideally, if one lab measures everything, things get easier, but we don't have this option. So yeah, a lot of time goes into this as well. So at the end, what we aim to do is to, broadly speaking, to minimize a cost function, which basically quantifies the difference between the experimental observations or the experimental measures and the model simulations. And what we aim to do is that the same model with the same set of parameters is able to go as closely as possible through all of the N experimental data sets that we have at hand. Here I just want to make a very small remark because of a question I got before this morning. So of course, there are also other methods to do this parameter estimation, for example, using Bayesian inference, and this is something that we have been doing lately as well. So another example of, this is the example of the final results that we get when fitting this particular model to all these different data sets. Another example is an infection with Mycobacterium tuberculosis. This is work from my master's student, Eliezer Flores. And what he did was he spent maybe half of his master's assembling this very nice and standardized database in close collaboration with our experimental group in the Institute for Nutritional Diseases, Rogelio Hernández Pando. So he constructed a model. In this case, it's only macrophages interacting with tuberculosis. So we don't have explicit representation of epithelial barrier. And then we ask, so this model includes also some quantitative information of rates of interaction. And we ask the model through optimization to go as close as possible to the different data sets shown here in these nice symbols for bacteria and macrophages. The last example in terms of calibration I will talk about is this SARS-CoV-2 infection model, which is being developed by Rebecca Pacheco, who is also my master's student. So I want to just emphasize here that instead of gathering experimental data, what she did was to do a systematic review, gathering clinical data, with which he then did a meta-analysis. And then we integrated our interaction network, wrote down the system of differential equations, and now he's in the phase of optimizing her parameters to have an idea of how different severities of patients can stratify through which kind of differential parameters. So the conclusion from this part one is that through the construction of mechanistic mathematical models that basically represent potential explanatory hypotheses of what could be causing or underlying a particular observed behavior, a disease mechanism, we can uncover a bit what is going on with behind the scenes. So here really the key is that we construct mathematical models representing these mechanisms, and we ask those models to robustly reproduce experimental data. So part two, once we have a model that reproduces a particular set of experimental or clinical data, now we can start playing with it. And the game we play is basically we hit it until we get a qualitative transition. In other words, what we want to look for is for bifurcation inducing perturbations. So we really want to understand what is the vulnerability of the system that gives rise to this qualitative transition between a healthy and a disease state, and probably also, hopefully, also back to a healthy state. So in mathematical words, what we do here is to really explore how the parameter space maps to the phenotypic space. Well, parameter which corresponds to the phenotypic and to the genotype and environmental space, and we really want to understand in mathematical terms these functions. So how movements or perturbations in this parameter space in given directions give rise to deviations from a setting point or a nominal behavior or phenotype in the trajectory space. So I started in one of the first examples I gave you. I showed you that I highlighted in circles that sometimes we already know beforehand what could be the potential bifurcation parameters, the potential risk factors, for example, by genome-wide association studies, but sometimes this is not clear at all. For example, in this model of airway epithelial infection, as is also in the case of COVID infection, we don't really know why the vast majority of us don't get any significant problems when we are exposed to this pathogen, but a small proportion of the population get very severely diseased. So this, in this case, we really want to understand what causes this dramatic shift between a very robust, healthy phenotype and a very, loosely speaking, unlikely but very, very bad course of the disease. So one of the things that we can do once we have a parameterized model, we can run, for example, a sensitivity analysis by adjusting the output function of this analysis to correspond to homeostasis, for example, to a measure of homeostasis. And we can rank them in terms of what are the parameters, meaning the kinetic interactions or the regulatory interactions in our system that most likely will account for the loss of homeostasis. So here we can, in this example, which is for airway epithelial infection, we were able to rank among, I don't know how many parameters they are, but definitely too many even to count them here, and also too many to do experiments with every single one of them. But we can pin out like the three, for example, that more dramatically change or are likely to change the course of the disease, and then focus on experimentations on those three, which in this case correspond again to basal barrier permeability, a carrying capacity of the bacteria, so nutrient availability, and macrophage strength. And once we have the risk factors or the potential bifurcation parameters, either because we knew them already or because our mathematical model pointed us into this direction. So here I will show you results on epidermis. We can start doing a bifurcation analysis and basically ask what is the effect of those risk factors, which can be represented as bifurcation parameters, on qualitative transitions or a qualitative behavior of our mathematical model. So here I show you, I hope, I hope it's big enough. Yeah. Here I show, this is a two-dimensional bifurcation diagram where each of the axes represent a particular severity of a given genetic risk factor. So here is the magnitude of barrier permeability, which corresponds to changing values of our parameter kappa P. And here we have the strength of the immune response, which can be tuned by changes in expression in, for example, toll-like receptors or antimicrobial peptides, which is our parameter alpha I. So when we change them continuously, so the first thing that should point out is that actually we get a very large region of a healthy or control phenotype. The largest region is this, for us, which are disease hunters, boring one, is this gray one, control region. This is actually very good news and is always what we would expect. So we are robustly healthy. So in general, thankfully, we are unlikely to get disease thanks to these regulatory mechanisms. So this makes sense that the widest region is healthy. And then we can also explore what happens if we change only one of the genetic factors and we enter into either an oscillatory regime or into a bistability regime, both of which represent not very severe phenotypes. So in bistability, one of the states is as the one which is totally healthy, and the other state is as the one that is totally unhealthy, that we'll explain in a moment. Oscillating regimes can often be not even diagnosed because sometimes, a kid has a disease and sometimes it has a flare and then it goes away before you even start going to the doctor. So it's not a very severe phenotype. But when you change both of them together, you enter into this abrupt, really unhealthy regime where you have only one steady state where everything goes wrong. So you have a persistent response of the system that cannot be reverted whatsoever. So this is a clear case of synergism between risk factors where only when we have two of them together, we can get a very dramatic phenotype. But then we can also ask, okay, if we take these two sets of genotypes, so these two cohorts, what happens if we expose those virtual patients to pathogen loads, which would be like the third risk factor in our model. And we can quantify with our model, we can quantify what is the vulnerability of the system to this kind of pathogenic challenges by really measuring what is the minimal amount of pathogen challenge required for disease aggravation. So to turning on part of the immune response that is very hard to reverse. And here we can see, we can measure these things, and we can also see why we get this very decreased tolerance of genotypes to pathogens load. So these values are much lower than this one, which would be like the healthy counterpart. So once we have that, then we can go to part 3, and this will be very short, so don't worry. So once we have identified these different dynamical regimes which correspond to the different phenotypes or the fragmentation of the disease space by bifurcation curves, we can now look for early warning signals that predict abrupt disease transitions. So I will tell you about the dream we have here. So the dream is actually justified in classical bifurcation theory. So when we work with smooth dynamical systems displaying the most common bifurcations, such as, for example, saddle node bifurcations, we expect that before the system goes from a normal to a disease state, we have something that is called critical slowing down that has to do with changes in the basins of attraction and the vector field. And this in turn relates to changes in to dramatic increases in statistical indicators such as variance and autocorrelations. So this is something that is very well known. But what is not known is what happens with non-classical bifurcations, for example, as those that characterize our piecewise smooth models. So here we have something called boundary equilibrium bifurcations And the dream we have is that also this kind of boundary equilibrium bifurcations that fragment the disease space into healthy and different disease phenotypes can also be predicted by these classical early warning signals. So this is work in progress, but it seems that yes. So the only thing I want to show you here is that, so here we were exploring if this red danger region can be predicted by a critical slowing down. And I hope that you agree with me that you can see a very sharp rise here. Currently, we are exploring if this is also associated to robust changes in variability of our system. So finally, the fourth part is the design of optimal treatment strategies. And here I really like to use this picture because this is a balancing act in which we really try to maximize effectiveness of the treatments while minimizing unwanted side effects. And to do so, one of the first things we have to account for is heterogeneity. So we want to really first start by stratifying the patients and just say, well, this bunch might be able to respond, this bunch might not be able to respond. And one of the ways we have addressed this in recent years was to adjust our mathematical model of atopic dermatitis. I talked to you about the skin model to patient data to achieve this patient stratification. So basically to look, for example, which particular patients would be good responders for a particular treatment. So once we have this stratification based on, again, this very strong interplay between modeling and fitting and adjusting to data, we can now start asking, well, what about treatment design? And one of the treatments that we were looking at early in this work was emollient treatment, which is a very simple cream that you just apply to the skin, and there were non-conclusive claims made that this would robustly prevent the onset or the risk of getting skin diseases. So we did a spatial model to systematically explore, or a spatial version of our model to systematically explore the effectiveness of different types of emollients in preventing disease. And in turn, our model suggests that it can be prevented regardless of genetic background. We can also go to the very severe phenotypes and try to use, use optimal control theory to design the best possible, so the minimally invasive trains of treatment, in this case corticosteroids together with emollients, this cream, so that the phenotype is robustly maintained in a healthy looking range. I say healthy looking because in contrast to a healthy system, a healthy looking, so in mathematical terms, it would be like an inverted pendulum, right? So it's a steady state, but that is not stable. So really have to constantly put control effort to stabilize this apparently healthy system. So it's these patients have to have this kind of treatment for a long time, maybe forever. So it's really important to minimize or to optimize the dose regimes. And we can also use bifurcation analysis to assess global effectiveness of treatment combinations, in this case, again, for atopic dermatitis for all different patient cohorts. So this is my favorite approach. I really like this kind of bifurcation analysis perspective, because we can really take all the possible genotypes or all the possible parameter combinations and see in this wide phenotypic space what is going on. And more formally, what we look here, for example, is we look, so if this is the bifurcation, the 2D bifurcation plot that I showed you before with these 4 phenotypes and this boundary equilibrium bifurcation curves separating them, we can then ask how all of these genetic variants respond to a given treatment. And the way we can do that in a mathematical way is to really look at how this boundary or these bifurcation curves shift. So this topology of change in the bifurcation diagram. So with this, we can answer things like how a given treatment affects all the possible virtual patients in a cohort. And we can also ask the reverse question. So we can fix a genotype, fix a patient, for example, pick this one here, which very severe symptoms, and now apply all the possible combinations. In this case, the treatments are the bifurcation parameters, and see what and describe the curves which correspond to all the combinations of optimal solutions to drive this person from severe disease to a healthy phenotype. So I am almost finished. I just want to say one more very last thing. So today I've shown you basically two main examples. I've been jumping back and forth between epidermis and airway epithelium, but in my group we study actually several other systems as well. And another dream that we have, I know we have many dreams, but it's is we want to ask if we can really make like a distillation, if we can abstract what are all the core and essential shared features in all these epithelial regulatory networks that account for a robust maintenance of homeostasis. And this is work in progress together with a very bright dynamical systems collaborators back in Chile and in Mexico, where we want to propose the simplest possible multi-scale network motif that can give rise to this kind of boundary equilibrium bifurcation transitions in response to genetic or to parameter variations that correspond to this balance between positive and negative feedback strengths. So with this, I come to the end of my talk. So today I have talked to you about how we use mathematical models to understand what are the mechanisms of change going from a healthy to a disease state. We have used mathematical models to ask which combinations of perturbations can be tolerated, and crucially, which one cannot, and how is homeostasis lost. We also started asking if early warning signals can predict such an imminent catastrophe, such as abrupt change in phenotype. This is still work in progress because of the non-classical nature of the bifurcations we work with. With this, we also ask if we can prevent this dynamic trajectory. And we have also used some modeling approaches to design optimal intervention strategies that look for minimizing negative side effects and also costs. So basically going back in this disease space. So mathematical models, I think they're very useful to understand mechanisms, to predict effects of perturbations and interventions, to design optimal treatment strategies, and to prevent catastrophic shifts. So now I'm open for questions. Thank you.
Thank you very much, Elisa. Really, really cool talk. So I have a comment and then a question. So the comment is for the sort of early warning stuff. I just think that's a really cool idea, right? I mean, just thinking about it in terms of the sort of the way one models phase transitions in physics, and thinking about how, you know, the change in the free energy spectrum, like early changes in the free energy spectrum, might presage the, you know, the existence of a phase transition in a dynamical setting. I think it's a very cool idea, and I think I'm interested to see where that goes. The question I have is for the earlier parts, for the homeostasis stability, you know, phase plots. You showed an example where, you know, you had sort of two clear properties that were the sort of main contributors, and then obviously the pathogen load as a third. But, you know, for these complicated systems and complicated regulatory networks, one is of course not ever guaranteed that, you're going to have a clear 2, or you might have 3 or 4 or 5, or there might not be any clear cutoff. So how would you handle, either higher dimensionality or the inability to make a clear cut, in the parameters that matter?
Yeah, it's a very good question. Actually, we were sort of discussing that earlier this morning. So there are 2 answers for that. So first, specifically in skin, so something that is really cool is that, but also limiting, is that in fact there are many things, or a couple of things, that can be measured in a non-invasive way. So this is really cool because it means that you can take dynamical samples even in babies without harming. And in fact, and you can use, for example, the response of the barrier to a tape strip. It's called tape stripping experiment. And then you can quantify that and really group that into a phenotypic space. And it's up to, you can score this and other measurements like redness. It's more like clinical signs, and really group them. So people do things in this case like a scoring system. And you say, well, if you have this score, you have a disease. If you have this one, you don't. So this is more a question from clinicians. And for us, from the modeling perspective, it's always very nice. I really love this analogy of input-output relations, because you really have to think about, okay, maybe you have a complex network, as you mentioned, but only a couple of the elements can actually be measured. So we really have to focus on how a perturbation translates to these measured and clinically relevant variables. However, in terms of or longer dimension or high dimensional measurements, so for example, there is also a lot of effort in grouping gene expression patterns in clouds and trying to understand how we go from one part of this cloud to the other through this dynamical system. So yeah, it's a very important question.
Thanks.
So I think we have a question on Zoom. Pierre, can you turn on your microphone and go ahead?
Yes, thank you very much for the talk. I suppose my question goes in a similar direction to Carl's, which is, if you make perturbations to your system of ODEs describing your system, can you make statements about how the bifurcation structure changes or doesn't change? I guess I'm thinking of things like phase transition in statistical physics, where some of the details will change in general, but this phase transition might belong to a certain universality class, and so you can make very general, robust predictions in that sense.
Yeah, that's a very, very interesting question, and again, it can be addressed from both from a let's say empirical perspective and also from a more theoretical perspective. So actually, these changes in the bifurcation structure, which would be equivalent to a further bifurcation of the system, maybe triggered by a n+1 bifurcation parameter, is something that I am always very keen to explore. So sometimes I feel that I'm like this horror story writer, where I really look for the worst possible ending and try to understand how we get there. So in fact, when I started working on atopic dermatitis model, I really tried to keep on perturbing and finding more perturbations, more bifurcation parameters to get something even worse than what we got. And it was just not possible. So there were only those dynamical regimes. So this is why we are currently expanding this model to really try to account for even worst case scenarios, which correspond to further bifurcations of this bifurcation structure. And so the way for doing that is, again, like searching in the literature and talking to people, both in the clinical and in the experimental literature. So what could be the other possible drivers and how we can build them into the model, which maybe they were not present before. If it's a system where we haven't analyzed so exhaustively, then maybe we're missing something. So there might be other parameters that are acting as bifurcation parameters. But from the mathematical perspective, there are different ways in which we can exhaustively ask if what are all the possible bifurcation structures that our model can exhibit. And here I just want to take the opportunity to mention or highlight that we always try to use the simplest possible models, exactly because this allows us this really wide exploration of the possible dynamic regimes of the system.
Thank you. But in that case, I guess my question is, if you use the simplest possible models as an abstraction of an unknown but possibly very much more high-dimensional model, how can you sort of control whether your bifurcation structure approximates the bifurcation structure of the, let's say, the unknown correct model in the same way as, or to the same degree of approximation as the ODE model you use approximates the unknown ODE model?
Yeah, well, I'm not sure if I understood your question, but the sad story about any modeler is that all models eventually will prove wrong.
Of course.
But the good part about it is that when they prove wrong, so when there is something that we cannot explain, it's actually when the nicest insights arise, because it's like this observation cannot be explained by this model. So something has to be added, something has to be changed, probably a feedback interaction or another variable or whatever has to be added to account for this change. So this is also why we also start building from the simplest possible model and basically hitting the wall, adding something, hitting the wall, adding something, and really trying to work in this way. I'm not sure if that answered your question, but
I think it does. Thank you.
Thanks for the talk. So I asked myself, you have there a lot of parameters in your model. And at some point I learned that you can reduce the parameter space by using dimensional analysis. Did you do this so to get rid of some of these?
Yeah, that's a very good question as well. So we are always in this constant conflict between like going to more modeling and simplifying as much as possible, but still having something with which we can directly relate to empirical observations. Because of that reason, we normally don't do this. So we always try to keep all the variables and all the parameters in a way that they can be easily interpreted at any stage of the process, because we are constantly talking to clinicians or experimentalists. So it's always, it's always easier if it's a more transparent, even though more cumbersome relation between the equations and the system. But yeah, we have done that to ask very specific questions, but in general, we don't do that for that reason.
But you don't lose any of your parameters. You just couple them together, basically. This is the idea of dimensional analysis. So this might help.
Yeah, no, but we really want to keep like direct and also directly measurable interpretations. For example, kinetic rates and this kind of things that can be measured by these killing assays, for example, that I mentioned before.
Okay.
So I do see the question on Zoom, but first one from the room.
Thank you. Thank you very much for a very nice presentation. I apologize that I came in a little late, so you might have already mentioned this, but I'm very curious about what the underlying data set is for you to generate these models. If you wanted to port this mathematical model to another, not the cutaneous issue, but I don't know, liver disease or some other medical condition, what type of data set would you want to see? And two, the other question, which is also related to the data set, is what type of data is underlined for you to detect the oscillation state? Because usually when you have a medical condition that you test it only once. You don't see how it's oscillating through throughout the states, as you mentioned. So I'm just wondering how you actually see that.
Yeah, well, I think I can answer both your question in one. So if I could write a wish list of what I want for the type of models we do, I would like models in which, data, not models, data in which as many variables as possible are measured in the highest possible dynamical resolution in a quantitative way in vivo and ideally at the single cell level. Of course, that's not possible. So, I mean, sometimes for some specific questions, there are some data sets like that available. And also this is one of the reasons why I move back and forth between different types of epithelial tissues. For example, for the epidermis, it's extremely hard to get like this high resolution, highly dimensional data sets, but it is dynamic. So we have fewer variables relating to the first question, but they have a dynamic resolution and they are in vivo, which is great. But some things, so instead of, I mean, I could put my wish list in the door of my office, but it's not, what I normally rather do is to go to the experimentalist and ask which type of data they have, because depending on that is the type of modeling we do. So I've also worked with this type of dynamical, high dimensional, very nice resolution, even single cell data, but for other type of systems, for example, for mammary epithelial cells. So yeah, we have worked on that as well, but here what we lose is this in vivo coupling. So it's always a trade-off, right? And well, in the oscillations, well, your question about oscillations is also related to that. So ideally, we would always have, so I guess in my list of priorities, I would put in the first point, I would put dynamical data. If we have dynamical data, that makes things so much easier. And sometimes there are, for example, clinical trials for atopic dermatitis children, where they measure over the course of several months, actually, how their symptoms fluctuate. So in some cases, we do have longitudinal data, or in general dynamical data, and this is like the ideal case scenario where we could also detect these kind of behaviors, such as oscillations.
Okay, thank you. But more, not in the sense of what would be optimal for you to have, but what would be minimal for you to have, where you might have some, if you have 300 samples of patients that are taken throughout the day for 10 days, would that be enough for you to start a model?
It depends on the question.
Okay, thank you.
So we seem to have a question on Zoom. Lutz, please turn on your microphone and speak.
Yeah, Lisa, thank you for this great talk. So you mentioned in your fourth part that you're extending your models also to a spatial aspect, and you consider the depth, I think, into the tissue layers. But there should also be interesting spatiotemporal dynamics along the epithelial surface. So one could extend, I guess, the same mechanism into a spatiotemporal coupled model, maybe using PDEs, and especially the antipeptide mechanism versus the barrier damage mechanism. They may have different spatial ranges in which they act, and that then raises the question if a barrier is never uniformly intact, but may have scratches and some random perturbations anyways, whether the fronts that separate the damaged region in an even homeostatic case from the intact region, whether these fronts would shrink the damaged region or would expand the damaged region. So that could be a different type of bifurcation, and therefore also two-dimensional response diagrams and so on that come from these variable spatial extents of your feedback loops. What is the approach that one could then use?
Yeah, that's a very good question. And actually, it's something that we have been thinking about very early on, but it's not, so this spatial aspect is interesting in many respects. So what you mentioned is like on the surface, like the horizontal, let's say, spread of the disease, if it would be someone lying and flat or whatever. So this is interesting in also because dermatologists very often assess patterns like redness patterns and patchy patterns, and really this spatial heterogeneity might be something very interesting, or is something very interesting specifically in dermatology research, because this is what people very often assess. However, we have not looked at that because our core interest, although it is a very interesting question, no doubt of that, but my really big interest, because I really want to understand how things can go terribly, terribly wrong. I want to understand eventually how a disease that originated at a particular epithelial tissue propagates to other organs. So more like vertical spatial dimension, so to say. And this is why our, in quotations, spatial considerations are accounted for rather in compartmentalized models. So eventually we would like to understand things like how inflammation in one tissue can spread to another tissue and predispose there, or decrease or increase the risk for disease somewhere else. But yeah, it's spatial aspect definitely interesting.
Thank you.
Questions? Then I think we should wrap it up here. And those who are still interested in meeting the speaker, there may be an opportunity tomorrow morning at breakfast. Just let me know, please. And then thank you for the talk, Elisa.
Thank you so much. There are some comments. Cannot hear. Audio not working. Sorry.